last but not least, we will end off with genetic engineering. Now just get everyone buzz with your neighbor. What are some of the key ingredients or starting points for the environment to be able to carry out natural selection on the population? Okay? Quick buzz. As we recap, what are the key raw ingredients so that natural selection can take place? Okay, let's start. Sarah, what is one of the starting points of natural selection? What do we need so that natural selection can take place? Okay, we need a pressure of some sort. The word you can use is selection, pressure. This pressure will exert some sort of a selection on the population. But there won't be selection if there isn't. Sorry? But before the selective advantage, there must have been something else. Okay, there must be a mutation, because mutation leads to. But mutation doesn't necessarily lead to advantage. Right? But it at least gives us a variation for the environment to play with. Some of the mutations may be advantageous, some may be disadvantageous. Mutations are neither good or bad. It is depending on the context of the situation. So, other than the selection pressure, we need random mutations that can lead to variation. On a genetic level, we are really talking about genes and alleles. Every allele is but a variant of the original gene. Just like how blood type O was the original. What now people say goat? G-O-T. I never understood why people say G-O-T, G-O-A-T all the time. Yeah. Greatest of all time. Yeah, then I was very confused. Sometimes I was offended. They will like say, oh, Lee Kuan Yew is the goat. And I'm like, what? What does that mean? Okay, so blood type O, the original. A, B, they are all alleles, they were variants, arose through mutation. With these two at stake, then natural selection can take place. Because as mentioned by Xiao Yang just now, some of these traits or alleles may have a selective advantage. What exactly is this advantage we're talking about? We're really talking about your advantage to be able to survive and reproduce. Because the moment you can do these two things, you can pass on your alleles to the next generation. And with enough time, repeat this over many generations, you'll find the proportion of individuals in the population having the allele will increase. And if your understanding of natural selection is strong, or at least you know the basics of it, you will then appreciate the idea that individuals, populations do? Over time. Individuals cannot evolve within your own generation. That's really metamorphosis. Or it could be the environment resulting in you changing. If your, for example, that trait is controlled by many genes, continuous variation, then the environment will exert some sort of a change on you. All of us were once babies. As you grow up taller, that's not evolution. And it's very important that we go away with the idea that the variation stems from mutation, not from a need or one thing to solve a problem. It's not like the ancestors of giraffes stretched their neck to the point that it became long. Then when they reproduce, they pass that stretched neck to the offspring. It doesn't work that way. To say that is as good as you all go for ear piercings, right? Then after your baby come out 2 holes on the ear. Cut your hair short just before having a baby. Baby come out, it grow up, can only reach that length. Cut your nails just before. So it doesn't work that way. It stems from a mutation. But actually, the scientific community used to think that it worked that way. That whatever you do in your lifetime will be passed on to the next generation. If you lose a limb in this lifetime, your baby will come out missing one limb also. Right? But it doesn't work that way. It's not in your genes. Therefore, at what point did the mutation need to take place? If, for example, you as an individual stay in the sun too long, UV rays keep hitting your skin and you get skin cancer, that be passed on to your little kids. So I'd like you to think that if the mutation needs to take place and needs to be passed on to the offspring, where must the mutation take place? When must it take place? Sorry? Your blood? Is your blood passed on to the baby? So when? When must it take place? What do you think? Straight a quick pass of the paper. Let's try to figure out when must the mutation take place? Because the counter example is you get skin cancer, we pass on to kids. You yourself want to send a son. Why is it not hereditary? If the cells of your skin, the DNA change, right? How come it's not hereditary? Okay, Dr. Me just brought up a question. You stand in the sun rays strike, UV rays strike your skin cells, they get mutated, skin cancer arises. But then the next question is, will that affect the sperm and eggs? Okay, ultimately, the mutation needs to happen when the sperm and egg is formed in individuals. Right? Start off with a single cell. If there's a mutation, and when they fuse together, this zygote that is formed will have that mutation now. Unless, of course, this person was sunbathing naked in the sun, then UV rays cannot penetrate that deep. What may be the cause of the mutation then? Usually it's during the copying process. During S phase, of meiosis, when they are copying DNA, cells also make mistakes. There can be copying errors. Actually, that's also one of the reasons why, as individuals grow older and older, the chances of an offspring coming out with a lot of genetic mutations increases. Because your ability of your body to copy things correctly gets worse and worse and worse as you grow older. Okay, so that's a quick recap of natural selection. This morning we're going to dive into another form of selection. The task for this morning I'll require is for you to, number one, you're going to watch this video. But as you watch this video, I'd like you to jot down in the points below what similarities and differences did you see between this form of selection, artificial selection, and natural selection. Are there similarities? Are there differences? That's what we're looking at this morning. And I brought to class an example of artificial selection. Sounds like your LA teacher. You cannot hear me from 4-7, right? No, there was once, right, I teach at JH403 at the other block, right? Yeah, I teach at this floor, right? Someone from the top floor on the other end, right, said that you can hear my voice. But your LA teacher, so crystal clear, I hear what he's teaching. You ready? Yeah, so differences, similarities. What's your LA teacher's name? Mr. Ahmad. Fun guy.
Selective breeding is also known as artificial selection. It is a process by which humans breed plants and animals with particular genetic characteristics. We have been doing this for thousands of years, ever since we first bred food crops from wild plants and started domesticating animals about 10,000 years ago. This is when we made the transition from hunter-gatherers to farmers. We no longer needed to wander around to gather food supplies. Wheat, barley, lentils, peas, rice, and potatoes were all cultivated by early citizens. We also began to tame animals for milk, meat, and hides. Goats, sheep, and chickens are thought to have been the first animals to be domesticated, followed later by bigger animals like horses and oxen for plowing and transportation. Agriculture allowed fewer people to provide more food. Regular, predictable food production led to increased populations and density of people. People now had the time to travel, trade, and communicate. Settlements began growing. The first villages and then cities were built near fields of domesticated plants. Civilizations founded upon selective breeding. How does selective breeding work? Parents with the desired characteristics are selected from a mixed population and are bred together. From the offspring, those with the desired characteristics are then also bred together. This continues over many generations until all the offspring share the desired characteristic. Just think of all the breeds of dog that are around today. These were all selectively bred from a common wolf ancestor. The desired characteristics that we select for can be chosen for usefulness or appearance, such as insect and disease resistance in food crops by breeding from the naturally resistant individuals to increase yields. Animals with white tiger is a good example of severe mutations boarding up due to the inbreeding of individuals that are too closely related. We have been selectively breeding for thousands of years, getting closer to our desired goal with each generation of offspring. Nowadays, we have come up with a faster way to do it: genetic engineering. We can transplant the gene for a desired characteristic straight into an organism. We will look at genetic engineering in more detail in another video. So there you have selective breeding. We have been doing it since the origins of agriculture and not looking to stop anytime soon. We do, however, need to bear in mind the potentially negative outcomes, such as the loss of genetic variety, potentially making the species vulnerable to other diseases.
Okay, some time now for you and your partner. What are some similarities and differences between natural and artificial selection? Do all these things still hold true for artificial selection? Are there similarities? Are there differences? Do we still require the same things? Okay, feel free to talk to your friends. Feel free to talk to your friend. Discuss. Once you're keen, you can view your friends' responses. Last minute, please submit your discussion points. Feel free to look at your friends' points as they are streaming in. interesting thing popping up. The idea of what advantage means. If you are done, scroll down, look at your friend's submissions. Okay, let's look at some of your friends sharing. Charlotte says, similarities is that you are passing on advantages alleles to the next generation. You look at the differences, artificial selection does not ensure that advantageous alleles for survival is passed on. Charlotte, just now I asked you, right, would you like to elaborate what is the difference between your first statement and second statement?
Like the first one, advantageous alleles for artificial selection means that the alleles they will be one, but then for natural selection is for the survival in the environment.
Okay. So Charlotte elaborated. She said that for artificial selection, the advantageous alleles is the one we want. The idea of advantage now is what is advantageous to us. But for artificial selection, the idea of advantages is being able to survive and reproduce. Okay? India says the similarity is that both of them require mutation in genes. Why? Okay. In both cases, so you say variation is needed. Be it whether it's the environmental pressure that selects or the human that selects, you need variation to be able to select. If they all look the same, they all have the same characteristics, there's nothing for you to select. Even the environment. The difference is that for natural selection, the selection pressure and advantage is determined by the environment. Whereas for humans, we decide what is advantageous. I'm going to scroll down and look at some of the new ones streaming in. Let's look at Nivita's point. Artificial breeding requires someone manually, but natural happens on its own. Okay, that's true actually. For artificial selection, humans literally step in. Later we will see an example. Natural selection takes longer than artificial. Oh, interesting. Why do you say that?
Artificial is you know what you want and then you can like choose one by one. And then natural you wait for a long time.
Right, okay. Actually, it was not said in the video, but actually it is true. For artificial selection, it is faster in the sense of the selection process. Yeah? But whether the organism reproduces fast, that cannot be changed. But what can be changed is the selection process. Yeah? Because you cannot control whether an owl will come down and eat the black mice or not. But you can control how fast you go and select what you want. That's an interesting point. Natural is for survival. Artificial is for aesthetic. Agree? Okay? Is it always for aesthetic? Okay, perhaps sometimes you want your animal or your plant to be more immune to a certain virus or bacteria, then you may breed for that. Is it an example of an aesthetic that you have in mind? Sorry? For dogs. You know dogs all, actually you can get any dog to interbreed with each other because fundamentally they are still one species. Yeah? They are still dog, but it's just that they look very, very different. Why do they look so different? Because humans have been artificially selecting for certain traits they prefer. But actually they are still the same species. It's a what, dog? The the ancestor of all dogs are wolves. After wolves got domesticated by humans to be more tame, from there on, they started breeding and selecting for certain traits that they prefer. In the population, there may have been some dogs that the body, okay, assuming this is the ancestor dog. All my dogs look like that. Okay. Yeah? Hey, the nose is much better. Yeah? Okay? The dogs, there's variation, but some people like certain variants more than others. Can you imagine in the population, original OG population, there may have been a dog that was just like this, that was smaller, born smaller, they're like 8 big chew. Let's take all the small ones and name each other. Then you end up getting all the chihuahuas, tiny little dogs. Then there may be some dogs, right? They came out a little bit longer in body. Then they find those pretty cute. They just kept taking those and breed each other, then you get longer and longer and longer dogs. So at the end of the day, we were selecting for certain traits. But actually, they have not evolved to the point where they cannot reproduce with each other yet. If organisms evolve to a point where they cannot reproduce with each other, they have become a totally new species. Just like how we cannot make chimpanzees anymore. Can is can, but no baby will come out. Oh, okay, Nivita says both require some sort of breeding. Why do you say some sort? Oh, it's not natural. How so? I see. Okay, got a point there. I don't think they forcibly like get the sperm from one dog and then forcibly put into the female. I don't think that happens that way. You just keep putting the dogs of the similar nature together in the same pen house. Then hope that something happens. Let's look at some other points. Let's look at Yuan's point. Yuan wrote quite a lot. She said similarities, genes and alleles are passed on to the next gen. Yeah? You need reproduction. Differences? No random mutation and variation. They are actively searched for. Oh, are you referring to artificial? Okay, so they are actively searched for. So what do you mean by they are actively searched for? Yeah. Right, okay. Okay, I agree with Yuan. The selection by humans is not random. We like the long one, we like the small one. But fundamentally at the core of it, what resulted in this dog giving birth to this one, and this dog giving birth to this is still random. It's as random as it gets. But the choice of us is not random. But mutation is always random. You actually cannot control what will come out. Selective advantage is having the phenotype the breeders are looking for. That's true. And we look for the most random of things. You see the breeds of dogs that exist. Some of them, I don't know how they can still exist if left into the wild. In fact, if you put it back into the wild, maybe natural selection will beat them out. Let's look at one last one. Let's look at Sarah's point. Both pass favorable, likable traits to the next generation. Is that true? In natural selection, do we pass on likable traits? But it could be favorable. What may be favorable? No, sorry. What may be likable may not be favorable for the organism. Possible? I may like that they are small, but they may not be favorable back in the wild. Natural selection is dependent on election failures. Right, popularity vote. In the environment, for artificial selection, it is determined by preference of humans. Yeah.
Humans are the selection preference.
Correct. So now, if we think about it, in artificial selection, if we match it, is there a pressure? That pressure are humans. humans. 407 said it is the human's desire. Humans, that is not really a pressure, right? Some traits don't necessarily confer a better chance of survival or reproduction, as we will see in a while. Do we still require mutations and variation? We still must have this. Without variation, there's nothing humans can choose. Is there always an advantage? Something very interesting brought up. What is likable may not be favorable. Is what is favorable always likable? Maybe not. A very big dog may not be likable by everyone. Okay? So this one, is there always an advantage? Not necessarily. Because sometimes we look at aesthetics. Sometimes we look at what is favorable to humans. Not necessarily the animal. Is this a say? Yeah. We will pass it on. I'm going to show you some videos now. I want you to, you think, is artificial selection right or wrong? LA. Don't can argue, huh? Is it right or wrong? Is it ethical? Anyone here has dogs? Okay. Is this the breed of dog you have? Pups? What? What's a chow chow? Okay, wait. Let's just Google it. How do you spell chow chow? C-O-W. It's not a cute name. Chow chow. Oh, wow. Okay, it's bigger than I thought. I thought chow chow sounds small. Oh, it's huge, huh?
It's from China.
Wow. Sorry, is it big? It looks big.
It's quite big, but there are some other chow chow can grow like really big, but mine is not that big.
So how big is your chow chow? Is that adult size yet?
Yeah.
That's adult already. It's like a tiny bear. These are, I think they are bulldogs.
Everyone in this story does too, but they're facing a health crisis and no one can agree how to save them.
I love French bulldogs because they're the most amazing dogs I've ever had the pleasure of meeting. Come on, baby girl. You speak to any French bulldog owner, and most of them have some kind of issue. Karen is my name, Karen Anderson. I have two French bulldogs. Stella is 3 years of age, and Boris is 16 months. Stella has had breathing issues from the day that I picked her up from the breeder. And right from that moment, I was told, even by the breeder, that's just French bulldogs, that's what they do. So it's pretty stressful when you see your dog, he wants to play, wants to run, but every time she does, she needs to stop and vomit.
Stella ended up needing a tracheostomy. She now breathes through a hole in her throat, can't bark and can't smell.
Stella cost me $4,500. First round of airway surgery was $2,000, and then the tracheostomy, that was a further $5,000. So, very expensive dog.
So we
See how
have
flat the face is?
every dog that's flat-faced has some degree of compromise, and nearly all of them benefit from some level of corrective surgery. These little guys, like, it's so overcrowded that the teeth become infected, and they often don't erupt properly. And if you don't get them out, they form a cyst, and literally it breaks, but it doesn't even crack as it breaks because there's not enough bone to even crack. It just falls apart. It's devastating. Something as simple as a trip up to the coffee shop, going to the beach, can be enough to overeat these dogs. People didn't know what they were doing is dangerous. They come in collapse and they're cooking from the inside and their organs are overheating. And there's some that you can save in the ICU, but there are lots that you can't. They've gone too far. We don't want that to happen to any more families.
The Australian Veterinary Association.
So what is likable here? The flat face. The flat face is not necessarily favorable to the organism. Because a flat face means you have less space. You squeeze a lot of important things into one flat piece of paper. Cannot eat that well, overcrowded teeth, they have to go through surgery to remove the teeth so that there's space before the jaw cracks. Not breathe properly, a lot of complications arise. But breeders still do it. They'll still breed to get the flattest space possible. Also don't know for what. Here's the same thing done on cows. Okay, not to get a cow with a flat face, but the humans have artificially selected for a particular trait that they like. Not marveling. This is for aesthetics also. Or aesthetics, not for food. Yeah, not for food, purely aesthetics. Okay, let's check.
In the cow
Cow.
He's quite big, yeah? He's 160 meters tall, yeah.
If I saw a man like that,
Yeah.
but I wouldn't get as close to him, I think. I would think he was on steroids or something. I think he was a bodybuilder.
It is 100% natural, whereby it's within so many generations, all breeders have taken the best of the best, or the more muscle of the more muscle. We move from a normal classical type of animal, and then we select that sort of animal based on the double muscle.
And when you say double muscle, do you mean it has two of every muscle?
No.
Because it looks like it does. It
It has exactly the same type of muscle than any other breed, except that in terms of quantity, each of these muscles is by far more developed.
Right. This cow is technically known as a Belgian Blue, and it's the product of selective breeding.
Selective breeding is the first stop on our tour of how man is using science to control nature. It's used by farmers to enhance desirable characteristics in their animals. In fact, selective breeding is all about managing sex. To create these Belgian Blues, over 100 years, farmers have only allowed the cows and bulls with the greatest muscle mass to mate. And the result is a bull that weighs over a ton. The animal's breeders are keen to show off the results of their hard work. The bulls are shaved to best display their muscles.
What they try to do is to treat the animals in order to highlight all the muscle. You see?
So you can see where all the meat is.
Exactly.
But even here, where they've sort of trimmed in there.
Yeah, to highlight that muscle, which has got a high value. You see?
And these are all going to be lean cuts, and they're all for quick cooking, are they?
That's right.
Because when you look at this, you can't help thinking of lunch, you know? Is it?
So what we can, should be, believe it or not, we've been doing it for really, really long. A lot of the things you eat have been artificially selected for to its modern day what you see. I'd like to show you two plants over here. Not plants you can find it in the supermarket. This is called kailan, it's called broccoli. They are the same plant. Yeah? They are the same plant. Just artificially selected for to enhance certain characteristics. The kailan has been artificially selected for so that we have more bigger leaves. The broccoli has been artificially selected for so that we have a lot more flower buds. But if you look at these two plants, as I'm going to pass them around, you find that the leaves of the broccoli look very much like the leaves of the kailan, just enhanced. You can in fact see the flower buds of the kailan, they look very similar to that of the broccoli, just enhanced. There are a lot of other plants that you eat that are the same plant. That means if I try to breed this and this together, they can breed because they are still the same plant. They have not become separate species yet. Okay, pass them around. Okay, let me blow your mind. So these are the other plants that are still the same species. Not chocolate. Broccoli.
No, it's not.
Alright. Okay, these are the other plants that you may have encountered before. So the likes of anyone eats kale? Here is the more extreme form of kailan. The leaves even bigger. If you select for, okay, so you see all these words: terminal bud, flower buds, roots, stem. So every plant, the OG plant is the wild mustard plant. Every plant has these parts. They have a stem, then they have the roots. They also have leaves. And I don't know if you notice, but usually at the leaves area, there are small little buds coming out. These are where new leaves can form. At the very top, you will also find a bud where new leaves can appear. You will also sometimes find flowers. So, this was the OG plant. Humans like the variant. They had more flowers. They kept selectively breeding for that. You get broccoli. Humans like the leaves so much that when they found a variant that was bigger, they kept breeding for that. You get kale. If you select for the roots, those that started the variants where the roots are slightly bigger, you just keep selecting for that, you end up getting a turnip. You keep selecting for this small little bud on top, you end up getting a cabbage. If you select for all these buds at the side, you end up getting a Brussels sprout. A lot of vegetables you see in the supermarket, they are just one and the same. Just selected for over many generations to enhance a certain trait that we prefer. Don't believe it if you look at the broccoli and the kailan. Actually, they look quite similar, just enhanced. They are, but this is like the dot of the plant of vegetables. Yeah? Yes. Okay, they all provide the same nutrients. This is the interesting thing. As we select for traits that look, as we select for physical, observable traits, actually one thing we do not account for is the inside nutrients. We don't know. We don't know if selecting for these variants, right, necessarily comes with better nutrition. Not sure. But some people suspect that this might be one of the reasons why the nutritional value of foods seem to be going down. Many fruits and vegetables have been domesticated and artificially selected to its current state. Artificial selection fruits. Don't know if you've seen this video before, but you can see the original form and the modern day form of fruits, certain fruits.
This is how Jane and Alfred
From watermelon, bananas, we have been doing it for hundreds of thousands of years.
You may take the appearance of everyday fruit and veggies for granted, but did you know they haven't always been what they currently do through selective breeding over the years? Here are 10 fruits and veggies that looked very different before we started cultivating them. Number 10, corn. The evolution of corn is a great example of how it can significantly change a vegetable over time. Corn actually comes from a Mexican grass called tisenti. The barely edible tisenti is small, skinny, and it has very few kernels. Back then, these kernels were also hard to reach since they were encased by a hard shell. To peel it, you had to hit it with a hard object several times. It's also speculated that it had a very dry taste, like a raw potato. The link between corn and tisenti wasn't very easy to find, actually. For many years, nobody knew where corn came from since it doesn't look like anything that grows in the wild. It was only after finding several similar chromosomes between the two that this link was determined. One scientist even managed to pop a tisenti kernel. The cultivation of tisenti to modern-day corn came about as, for each subsequent harvest, we selective bred the more desirable tisenti plants that had larger, more rows of kernels. This led to corn we know today, which is easy to peel and 1,000 times larger. Modern corn also contains 4 times more sugar compared to the natural old version of corn. Number 9, avocado. The millennium's favorite fruit has gone through quite a lot of change, and it's all for the better. In the wild, avocados are very small and can easily fit into the center of your palm. Growing to about 3 inches in diameter, the pit of the wild avocado takes almost all the space inside the fruit, leaving little room for the fruit's famous green flesh. Roughly speaking, it would take close to 10 wild avocados to get the same amount of flesh that you would get with a single modern version of the fruit. Worse yet, the little flesh that it has doesn't
doesn't taste very good. It has a rougher, grittier quality, in contrast to the creamy and tasty characteristic of the modern avocado. The wild avocado was also encased in a hard shell, quite different from the leathery, fleshy kind of skin that the
Okay, many other plants. Actually, most of the plants you see in a supermarket have been selectively bred. Okay, so we have come to the end of artificial selection. We can do it, but should we? Because scientists have found an even faster way. Why we skip the breeding process and we just take out the gene that we want or the allele that we want and just directly put it into the organism. It's much faster one. You don't have to go through the multiple generations. We search through the DNA, find what we want, take it out and put it into the organism. That's what's faster. And that is the last part of the chapter. Genetic engineering. We engineer our own organisms and plants. You all do this in LA? Yeah. So you all roughly know this, huh? Genetic engineering. You will know the broad idea of genetic engineering. Broadly speaking, if you were to hear someone say genetic engineering, they usually are referring to if there's organism 1 and organism 2, we are trying to transfer things that we want from A's DNA to B's DNA. And if B goes on to multiply or grow up into an adult, then we have a multicellular organism, all with the transferred DNA. That's really the gist of genetic engineering. We skip the multiple generations and time needed. We just go straight to the point. Is this dangerous? Actually, hard to say. Because we cannot tell whether inserting the gene into a foreign gene into an organism can mess up their biology or not. We are really not sure. And that is one of the biggest contentions of genetic engineering. But for us, I'd like to focus on how we can carry out genetic engineering, the science behind genetic engineering. In this last part of the lesson, we're going to look at how we can carry out genetic engineering. By the end of this section, by the end of this first section, I'd like you to be able to identify the tools required for carrying out the process. I'll be introducing certain tools that humans use to transfer alleles from one organism to another. Oh, how? What? You all get the word plasmid? Okay. Got my hopes up. Okay. After you watch this video, we'll end up with an activity and we're going to try out genetic engineering ourselves. Okay? Using paper. Okay.
The first step in cloning a gene is to isolate.
Okay. If you hear the word clone a gene, it means to take it out of the original organism. By taking it out of the original organism and putting it into a new one, we essentially clone the gene. Yeah? We make a copy of what it was in the original organism. Okay, so that's why if you hear the word gene cloning, what we mean is that we are creating a copy of what was in the original organism and now taking it out. Okay? So we're going to learn the various tools required for this process. Take out the gene and to put it into a new organism.
First step in cloning a gene is to isolate the DNA from the organism that contains the desired gene. The isolated DNA is purified and then fragmented with a restriction enzyme. Restriction enzymes used in cloning produce staggered cuts in specific sequences in the DNA, generating fragments with cohesive ends. Each fragment has a single-stranded sequence of nucleotides on its ends that is capable of hybridizing with DNA that has been fragmented with the same restriction enzyme. The DNA fragments are then incorporated into plasmids. The type of plasmid used for cloning has a single restriction site, and when cleaved by the restriction enzyme, generates the same cohesive ends that are in the fragments of the DNA to be cloned. The cohesive ends of the plasmid and DNA fragments now line up, and the enzyme, DNA ligase, is used to form phosphodiester bonds. The next step is to incorporate the plasmids into bacterial host cells by transformation. Each cell contains a different segment of DNA from the original organism. Taken together, these cells represent a DNA library. The cells can now be plated out on an agar medium. The colony of cells containing the desired cloned gene can then be identified and isolated.
Supposing this particular bacteria has the gene we want for maybe resistance, we can take this bacteria, let it grow out in numbers, then after that we extract out the DNA, then we put it into whatever organism we want to genetically engineer. So that's why we call it a library, except we don't use books to store the information, we use bacteria, each bacteria storing different fragment of a gene. So you have a question? Okay, there are many ways to put it in. Which will not be the scope of our last chapter. Some people use a gene gun. Yes?
Control like how they break off, or is it just random?
Okay, you in a sense can control using enzymes, which is what we're going to look at this very last part. Restriction enzymes. Restriction enzymes, think of them as scissors. Yeah? And we have many kinds of scissors that can cut at different points. So if we want a particular gene, we look for where this enzyme can cut so that we can extract out this gene. We're going to try it out in a moment's time. These are the tools that are required to carry out genetic engineering. Number one, you heard that there are restriction enzymes. These are like scissors. Number two, you hear that you also need a gene of interest, or they put here desired gene. I'm going to say gene of interest. This is what we want. You also heard the word plasmid. You heard the word ligase. Okay, this process is really simple. The difficulty lies in just remembering the names. Okay, I'm going to show you the broad idea of this. Supposing you have an organism's DNA and the organism has a gene that you want. You heard that there's a plasmid. A plasmid is nothing more than a DNA that is circular in nature that connects end to end. DNA, plasmids are circular DNA. They come from bacteria. But humans love plasmids because plasmids are very good, small little packages that we can use to contain our gene of interest. Okay, so the process I'm about to teach you just goes really like that. We need to, number one, learn how to cut the gene of interest out. We need to learn how to cut the plasmid so that we can create a plasmid that contains the gene of interest. Why do this? Because this over here can then be transferred into any organism we want to modify. Let's say we want to transfer it to a fish egg that will eventually grow to an adult fish. We want to transfer it to a plant cell that will grow up into an adult plant. This is what we require. We require a package that contains the gene of interest. So that's what we are going to learn today. In this last part, we're just going to learn how to prepare this. And what is this called? This structure is called a recombinant because we recombine plasmid. It is a plasmid that has been recombined with the gene of interest. this is our last part of the chapter. We just want to know how to create a recombinant plasmid. This recombinant plasmid is very special. You can then use it and put it into whatever organism we want. So, we have scissors, we have plasmid. Last thing, ligase. Ligase, if we have scissors, we need glue. We need glue to be able to stick them together. Yeah? So, the tools required are actually quite logical. We need scissors to cut, and then we need glue to stitch it together. That's where ligase comes in. And these are the basic tools. Alright, now we're going to try it out. Sorry, got a question? You just what? Yes. Right. Right now we're going to try ourselves. On the screen, I am showing you 3 different kinds of scissors restriction enzymes. Each one of these restriction enzymes can cut at different points. Notice that the restriction enzymes don't cut just like snip like that. They actually cut zigzag. You see? They cut at a zigzag manner. The beauty of cutting in a zigzag manner is that you create something called sticky ends. It is sticky because if you were to use these same scissors to cut the gene of interest and cut the plasmid, the sticky ends will be complementary to each other. Then it can hydrogen-base pair. We'll try that out in a while, yeah? Okay, so the activity will go something like that. I have presented on the screen 3 different kinds of scissors restriction enzymes. Here you will find inside this cup noodle box lots of different gene of interest. The gene of interest I have labeled and I've also colored it black color. This is the gene of interest you want to focus on in a while. To the left and right of the gene of interest, there are sites where you can cut out. Okay? Later on, the challenge for you is when you receive your gene of interest, I'd like you to find out which scissors you can use to make your snips. Because remember, you have to make 2 snips first, right? To extract out our gene of interest. Sorry, I don't have enough paper, but this gene is supposed to be very, very long. Right? I still need you to cut out your gene of interest in a while. Okay? That's the first one. It's random. Huh? Every gene of interest has different sites where you can cut. Next, you need your plasmids. Okay? When you come to the front later and collect your plasmid, so I'd like you to do, take a slip of paper, just take one little bit of scotch tape, then you need to create a plasmid because it's circular, right? I've not done it yet. So you just tape it end to end. You see this white end over here? Then you should see a ORI. Later on, you just overlap and you just stick like that. Then you will create a plasmid. After creating a plasmid, I'd like you to search within your plasmid to try to find where you can use the same scissors to cut. I've actually underlined for you where you can find the region to cut. I've labeled it MCS. MCS. This MCS region is where you can search for the patterns where you can use your scissors to cut. So the goal for you is this. Later on when you collect your plasmid, your gene of interest, I want you to just be able to, number 1, tell me which enzyme you are going to use. After you've decided which enzyme you want to use to create sticky ends, if you have scissors, you may proceed to cut out sticky ends, you join them together, or you can carefully tear. After that, you can come back to the front and grab some scotch tape to tape your plasmid and create your recombinant plasmid. What your end product should look like, it should look like this. You should end up with something like that. Your gene of interest, where you have created a sticky end, and you should also have the original plasmid, but now joined together to form a recombinant plasmid. So really all I need you to pick up from today is to be able to identify where you want to cut, and identify which enzyme you want to use to cut. Maybe it doesn't make so much sense yet until you receive the item. Okay, so may I get one by one, starting from the other side? Come to the front, collect a gene of interest, collect a plasmid, and a little bit of scotch tape. And you may begin the moment you get it. I will go around to address any queries you may have until you can perform and create your recombinant plasmid. Okay, you can come. Okay, so random, just collect one. Okay, take a little bit of scotch tape. Oh, you just take that one. And then you go back. Alright, next row. Once you receive your long strip, create a plasmid first. Use your scotch tape, tape it end to end. Then look to the screen. Which restriction enzyme do you want to use to cut your gene of interest? Okay, next one. Okay, last column. I suggest before you cut, maybe use a pen to figure out where you want to cut first. Your cut should be a zigzag cut. Okay, before you make your cut, you raise your hand. Right, once you think you decided which one you want, where you want to cut, you raise your hand, I'll come to you. A double check. Yeah, I can roughly tell if you understand what you're supposed to do. nucleus of the task. Right now, you should have circular DNA that goes something like that, right? Number one, I want you to you also should have the gene of interest like that. Number one, you decide where you want to cut. Number two, you have to decide where you want to cut also, such that you can combine these ends to these ends over here. But the trick, the difficulty is that whatever sequence is over here and here needs to be complementary to whatever you cut here. If not, they cannot join to each other. Okay? Yeah. Sure. These are just three different enzymes that you can use to cut. Maybe everyone, you can look at your gene of interest first. Can you see to the left and right of your gene of interest, you see a sequence that is 6 nucleotides long? Can you see? Okay. Based on your site, which enzyme would you want to use, can you use to cut? Each one of you should be different. For example, my sequence, the two sides at the opposite ends goes GAA-TTC. That means I can only use ECO-RI to cut. Have you identified your enzyme yet? Identify already? Okay, so this is the next thing you can do. For myself, mine goes GAA-TTC, CTT, AAG. You will cut this way, make one slip down, make another slip across, then you make another slip down on both sides to create two ends that are sticking out. Okay, try that first. Can you create sticky ends? Okay, for those that are still very lost, I'm going to show you again. This is your gene of interest. You should find on opposite ends something like that. So for me, I got GAA, TTC, CTT, AAG. And I have another of the same thing on the opposite side. GAA, TTC, CTT, AAG. Okay, and then you still have other sequences, yeah? Your task now, just take scissors to cut down like that. Take scissors and cut like that. And what you will end up with will be something like that. AATTC, G. You have a gene of interest. And you should have GCTTAA. Sticking out like that. You will have a slip of paper with what we call sticky end. Okay? Create sticky ends from your gene of interest, step 1. After that, I would like you to search your classmate to find where you can create one more sticky end so that you can insert your gene of interest in. You know where to cut. Also, cut down vertically, horizontally, vertically. Down, there you go. Cut down, across like that. Yes, so you search on that level, find where there's the same sequence so that you can form the same part. No, those are just extra layers. Just need to find the correct sequence here. So yours is AGA. Let's try search here. You perform the same cut. Down, horizontal down. Now you can stick it. Now search over here on the NCS region. Oh, no, you just need a one cut. Okay. So you found what's the GA? Found GA. You cut vertically down, horizontally, and vertically down, right? Now you produce two sticky ends that you just slot in. Correct, the cut first. Once you cut it out, vertically, horizontally, and vertically. Yes. Yeah. Okay. So this combination so you just with one cut your classmate cuts to change. What is your sequence? Okay, so you found it. Right. A, because the movement A, G, C, A, G. Right. So you cut down. Then you end up with a zigzag, right? Now your zigzag, you alright, so technically now vertically horizontally. Usually must be that's why you must use the same enzyme. Use the same enzyme, then you create the same jigsaw puzzle that you can fit in. Okay, so you figure out the particulars. So, you respond to AAGC. AAGC. Found it. Then technically, if you were to cut down like that, pull it apart, you would have created two sticky ends. Right? And you can just connect it. So you would have created two jigsaw puzzles and just connect them. But that's fine. You only need one, two. Because the moment you make one part, you will produce two sticky ends. Right? Only one cut is needed on the pass. Interesting. I noticed a lot of you are trying to find two cuts to make on the plasmid. You only really need to make one cut. Because you make one zigzag cut on the plasmid, you end up with two ends that you can use to stick. Yeah. So it's really about creating the correct jigsaw puzzle to fit in. Yeah, it is still complementary. Then we go to the other side. Oh, you can get out. You understand now. So that's right. Okay, I understand. Okay, I'll need to end off here. It's 9:30. Okay, there's a lot of trial and error on our part, but notice the fundamental idea is you need to make the same cut. Once you make the same cut, you have the complementary ends that can stick to each other. It's really about just creating a jigsaw. Okay? That's the fundamental idea. Will I ask you to do this during an exam? No. I'll just provide you the sequence. You go and spot where you want to cut. Yeah? Okay? Okay, that's all for today. You can go for your next lesson, wherever you may be.